Okay, next hormone is about cytokinine. Okay. Okay, so normally cytokinine is synthesized in the root and transported to other organs, as although there are many minor sites of production as well. So the major synthesis site of the production of gibberellin is root. Compared to auxin, auxin is meristematic cell at the root and shoot. For cytokinine, it is produced in root, but actually other part also can produce as well, but the most important one is at root. The major biosynthesis of the gibberellin, cytokinine is in root. Okay, and then this is the general function. So normally the main function is to regulate cell division in shoot and root. Yeah, just now auxin is play the role in cell elongation, but cytokinine play the role in cell division. These are totally two different functions. Cell division and cell elongation are different. Cell elongation just increase the size of the cell only. It will not divide. It will not cannot produce 2 cell, 3 cell, 4 cell like that. It will not beranak. In human, you can say it tak akan lahir lah. Dia lahir anak-anak dia. But cytokinine, it will lahir anak-anak mereka. And then not only that, it will also modify apical dominance, promote lateral bud growth, and promote movement of nutrient into zinc tissue, stimulate seed germination, and also delay leaf census. But mostly we will focus about cell division and also the apical dominance. Okay. So this is the roles of the cytokinine. So it is act as the control of cell division and differentiation process. So normally cytokinine will be add in with the auxin one. It will like the abang-adik lah. Kalau ada auxin, memang ada cytokinine. They will collaborate to each other one. So cytokinine stimulates cell division and influence the pathway of differentiation. So normally when you have the tissue, when the plant cell is cultured in absence of cytokinine, the cell will grow very large but does not undergo mitosis. So this is the key point that I want to tell you. Meaning that auxin is only grow very large only. The cell number is still maintaining. What you place, how many cell number in the culture, meaning maybe 10 cell, you place 10 cell, then finally you only have 10 cell too. But just the cell size will very large only. But cytokinine will play the role in mitosis, cell division. It will produce large number of cells. So maybe you culture 10 cell, in maybe 1 week, 2 weeks, you will have 20 cell, 30 cell, or even 50 cell like that. Okay, so normally cytokinine can collaborate with the auxin. So the ratio of cytokinine and auxin can control the cell differentiation. So if you see this diagram lah. So this diagram I summarize into this lah. So when auxin is equal to cytokinine, meaning that it will induce catalyst. So what is catalyst? So catalyst actually is the plant is you can say that is one type of the plant structure that can play the role in further cell development. Meaning so in general, the plant in natural habitat, plant will not induce catalyst one. Normally this one is that human made in tissue culture technology. Normally this one is because of the human want to do the experiment about the plant. So if you want to do the experiment in the plant, normally we will not grow the plant as well like the farmer. In laboratory, we will induce the catalyst only, because this catalyst later can develop into leaf, can develop into shoot, can develop into and flower like that. All the plant structure can be developed from the catalyst. So it is very useful structure for the researcher to make a research about the plant tissue culture lah. So in natural, catalyst will not form in natural lah, very rare. It is very rare situation that the plant will form catalyst. But for generally, plant will either that the auxin concentration is higher than cytokinine, or cytokinine is higher than auxin. So when auxin is higher than cytokinine, it will induce rooting. So meaning that when the plants are exposed more auxin concentration, it will induce rooting one. Only auxin induce rooting. But if cytokinine is higher compared to auxin, it will induce shooting. So induce rooting mean that the growth of the shoot is very slow or even no growth, but the root will grow very fast. Opposite to cytokinine, when cytokinine is higher than auxin, it will induce shooting, meaning that the growth of the root is very slow, but the shoot growth will very high. So this is the meaning about the cytokinine. Okay, and then it also play the role control of apical dominance. So as I mentioned before, we know that auxin play the role in enhance apical dominance, and we know that apical dominance inhibit the growth of the axillary bud. So when you remove the apical bud, no auxin at all, cytokinine from the shoot antagonize the action of auxin, allowing a limited amount of axillary bud growth. So it will induce the growth of axillary bud. So the axillary bud farthest from the apex are increasingly elongated. So meaning that if although apical bud is still maintaining at there, meaning that it got auxin although you have the plant have the auxin yet, but the auxin but the cytokinine still can antagonize, it will counteract the action of the auxin one. It will still can stimulate the growth of axillary bud, but the growth is very slow, not very fast like this. This one is where the auxin is already totally removed, meaning that auxin level also decline, particularly near the gut surface, and then the decline topmost axillary bud in particular to go and take over the new apical bud. So I just want to mention about the cytokinine play control about the apical dominance. It can control the apical dominance one. So like that. Okay. And then another function is about anti-aging effect. So meaning that anti-aging, we know that aging is tua right, orang itu bahasa Melayu kami panggil orang tua. We know that in human we will have going to death phase kami akan jadi tua kan, 60 tahun 70 tahun akan jadi tua, lepas tu mati. But cytokinine can prevent the plant go to aging. So that is why some of the plant can grow unlimited, because got cytokinine. Cytokinine already play the role in cell division, and then it also can provide anti-aging effect. So that is why that some of the plant cannot die one, and then secondary plant will have the unlimited growth. Okay, so cytokinine slow the aging of certain plant organ by inhibiting protein breakdown. And then if leaf removed from a plant are deep cytokinine solution, they stay green much longer than otherwise. So this is the role of cytokinine